From Microbe TV, this is TWiN, This Week in Neuroscience, episode 58, recorded on February 24, 2025. I'm Vincent Racaniello, and you're listening to the podcast about the nervous system. Joining me today from New Orleans, Vivian Morrison.
Hi, guys.
Welcome back.
Thank you.
Missed you. Good to have you. We need your insights.
Fantastic. Glad to do whatever I can, or just, fill the airwaves.
Also joining us from New York, Tim Chung.
Hello, hi. Yeah, welcome back, Vivian.
Thank you.
Great to have you back.
I'm glad to be here.
Especially for any topic dealing with social neuroscience. Since your family is growing, there's a lot of socialness going on there, I'm sure.
Oh, it's true. No tongue dragging, luckily.
Not yet. Have you seen
yet.
your kids play fight? Maybe they do tongue drag.
Yeah, maybe.
Well, if you like these programs and what we do, talk about science, real science, not fake science, we'd love your support. Go to microbe.tv/contribute. Science is under attack, and we are here to try and repel those attacks. We need your support, microbe.tv/contribute. So in honor of Vivian's return, when were you last on? Was it like the fall of last year or November-ish?
September. Or maybe not even. When did y'all record in September? Because he was born on the 21st. Connor was born on the 21st. So I might have been out.
May not have been.
My memory is not nearly that good.
Mine isn't either. If you all recall, the last one I was on was about brain changes after pregnancy. So I'm living this.
Okay.
I'm living this.
That's
It's fantastic. It's so interesting.
So you can confirm that there are brain changes after
Absolutely.
pregnancy?
Yep.
Okay.
I know they actually said in the paper, I believe they were like, there's a time when, there's specific hippocampal changes that occur like during pregnancy, and then afterwards they kind of revert back. And I was like, there was a period there where I couldn't remember what had happened like 5 minutes ago. And I was like, yep, my hippocampus is not like talking to my cortex very well. So I'm not like, encoding or retrieving memories the way I typically would. So yeah, it was kind of fun to think about.
Is it correlated with a loss of spatial awareness as well, which is the other thing that hippocampus is supposed to be?
Right.
So you're
But the question is dorsal versus ventral hippocampus, right?
Oh,
because I think spatial tends to be more dorsal, and then the more ventral you go, you have more of these like social facial or social stuff.
Okay.
Yeah.
Okay. Makes sense.
Well, in honor of your return, you get to do the paper today.
I know. And it's equally as exciting as the last time I was on. Actually, it does have some kind of like conceptual similarities that I'll bring up right at the beginning. So should I get started?
Yeah.
Yes,
all
please.
Right. I'll say, like, I don't know, Tim and Vincent, if you guys were able to read it, but I really feel like this paper caught me on a good day, Like there are some days when I try to understand this stuff and I'm like, nope, can't do it. But today, like when I read this paper, I was like, oh, like I understand. And I think it actually has to do with having helped a colleague here review his grants. And they had a lot of the transgenic animals and viral constructs that we're going to talk about today were implicated in his work. And it's really just that like exposure and trying to like work out how these different tools fit together that really made it possible for me to understand what was happening in this paper. All right. So the title of the paper we're discussing today is Rescue-like Behavior in a Bystander Mouse Toward Anesthetized Conspecific, so that just means another mouse, promotes arousal via a tongue-brain connection. And I actually heard about this paper on the radio, on NPR, as I was driving home. And I was like, well, if NPR is doing something on it, then I have to do it. So picture yourself, you're walking through the office. Maybe you're going to get a cup of coffee from the break room, and you turn the corner and you discover that one of your colleagues is sprawled out on the floor, unconscious. What do? So you might rush to his side and like slap him in the face or take him by the shoulders and shake him to try to rouse him, which, by the way, you really should never do, because what if the person had like a traumatic spinal injury? You'd probably kill them. But in any case, you might do those things. But if you're a mouse, one of the mice in this study, then you might sniff him, groom him, lick his eyeballs, and most importantly, it turns out, pull on his tongue. Yep, we're going to talk about tongue dragging. So
pull on his tongue, but with their teeth or something like that?
Yes, with their teeth. So this tongue dragging.
Although, just disclaimer, as a human, you really should just call 911.
Yes. Yeah. I mean,
which
the first
is not
thing.
available from.
Yes. They with their little paws, they don't have like opposable thumbs, so handling tiny cell phones is difficult. They're always getting lost in the bedding, So the experimenters here, the authors of this paper, observed this tongue dragging behavior, and I don't actually know what studies what prompted this. Like, what studies were they doing?
They did mention. Well, it's actually quite interesting, because when I was growing up, people kept saying that the way to think about neuroscience and mouse and rat behavior, rodent behavior, which a lot of neuroscientists use as model organism, is not to put yourself. Don't think of them as tiny people. They really are just mice, and they are just machine. Think of them as very simplistic machine. They have no sense of self. They have no sense. They have no emotion. They have no empathy. So they can't really, they don't really help other mice out. They just, think of a very simple machine that think of themselves. They just need food, sex. That's pretty much all they need. But
no water.
Or water. So, yeah, exactly. Food, sex, water. That's all. A bit of a warmth. But then, since then, people have increasingly found more and more evidence of like rodents perhaps displaying behavior that's a little bit difficult to explain if they're just very selfish creatures. Like, for example, they have found if you lock a rodent, I think it's a rat, inside a cage, and then put a friend rat, so maybe they're cage mates or something, outside of the locked rodent's cage, and give the friend rat a choice between like chocolate. Maybe I'm making this up. Sometimes listeners should go and fact check me. But first of all, if you give a rat a cage mate that is locked behind bars, the rat would actually go and try to break his friend out of the cage if there's a way. So they would sniff the cage. If there's a door, they would try to they would mess around with it until it's open to rescue his friend. And apparently, if I remember correctly, if you give them a choice between like chocolate and rescuing his distressed friend, they would actually go for the distressed friend.
Well, an important question here is, was the chocolate milk chocolate or 78%? Because obviously if it's dark chocolate,
no one likes it.
But
yeah.
But I mean, it's they do mention this, and I mean, and they see it in other organisms as well, right? Like non-human primates, and I have no idea.
But for rodents to have a sense of, because rodents, anyone who's had, so rodents actually, it's weird that rodents can display this kind of behavior, because rodents sometimes, if it's kind of a little bit gruesome, but in mouse, researcher would know sometimes your mice can die of natural causes in the home cage, and the reaction of the cage mate is cannibalism. They would eat that mouse.
I was going to say, so I have seen this before, this behavior, and I was like, oh no, what's about to happen? I need to leave, because I was like, oh no, they're going to eat me.
It can also get kind of gross, but so it's kind of weird that there might be this kind of balance of like, if a mouse is still alive, then you would try to help the mouse, but if the mouse is dead, then as a mouse you just apparently cannibalize that mouse.
It's we used to see if you have a pregnant mouse give birth and there were stressful conditions in the mouse, the mom would eat the babies, and they're alive, so not always dead.
By little side note, I may have mentioned this really fascinating book called Eve. I may have mentioned it last time I was on, but they actually talk about why that happens, and that it happens in other species, like I think maybe there's some. Not humans, no, but they talk about how, like, it's interesting. Go check it out, folks. Eve, how the female body, changed 200 something thousand million years of human evolution. But yeah, it's these mice can sometimes do they do lots of weird things, and like you're saying, Tim, I think we're just starting to understand, or just starting to piece together what these mean for the mice. But
like mice helping each other out is definitely a new phenomenon, and they also mention it's like mice. If a mouse has been like shocked or like been experimented, sometimes do have
pain.
They experience pain, and then a friendly cage mate would go and quote unquote console the mouse. So they would lick the mouse that's in distress and stuff like that, which is, it's kind of hard to explain if you just treat the mouse as a simple selfish machine. And whether we want to put how much we want to anthropomorphize these mice is a huge debate, I think. But definitely these are like behavior that people have documented. And kind of the neuromechanism is something that we're going to talk about today. But maybe an also equally interesting question is like, is there any functional consequence? Like, did they evolve to do this, and does it serve a purpose?
Yeah.
Anyway, so all these social interaction is very interesting.
Yeah, and I think the authors did a really good job of not lingering too much on is it empathy? Are they trying to help? Why would they want to help?
Sure.
Like when on NPR, when they were talking about it, they of course they took the stance of like, oh, maybe the mice are empathetic and this like changes how we think about them, which maybe it does. But then, in order to kind of like emphasize that, they had somebody who was like, no, like it has nothing to do with that. It's just they discovered a cool behavior. And then somebody else was like, I think it really makes us question. But so that was NPR. But when you read the paper, it's like, they're like, maybe, but let's get into mapping the circuits.
Yeah, but it makes sense. Like even for humans, sometimes like you hear a baby screaming next door. You might not be very empathetic to the baby. I'm not going to put myself into the shoes of a baby. I'm just going to give that baby whatever it takes to shut that baby up. Yeah. So maybe something like that is the same with mice, with another mice in distress. It's making a horrible scream and you're just trying to get it to stop.
Yeah, because ultrasonic vocalizations are a thing. So there are, let's get into it, but I want to bring up 3 things that I hope everyone can walk away with from this episode. And the first is mapping circuits. So it's a neuroscience thing, I think, mostly. How do we do it and why is it so special? Like, I think we as neuroscientists, take this for granted. And it is immensely complicated, but really clever. And I love the way that they use these tools and how they map the circuits that are involved in these behaviors. And that leads me to the second point, which is you really, when you're doing good science, you should always be triangulating onto your hypotheses. So if X is true, when we do Y, we should see Y sub 1. And when we do Z, we should see Z sub 1. So you have to kind of rely on these multiple angles to really conclude something. And I think more and more, like, you have to do that. And, later on, I probably will talk about like beating a dead horse, but that's really what you have to do to make sure that there were no other ways that you could have, misinterpreted the data that you get from just doing one experiment that's done in one way that has its own caveats. And then the final point is that bigger is not always better. The size of the circuit, the number of neurons that get recruited, these things are not necessarily indicative of the importance of a circuit for a given behavior. So it may matter more how sensitive or well-tuned a smaller population of cells is to a given stimulus. And this is actually something that we talked about the last time I was on, and I talked about brain changes during pregnancy, that there was kind of an overall reduction in activity in a certain network, but then those cells responded much more quickly or more robustly to a very specific phenomenon, which was, I think, the baby crying or infant-related behaviors or stimuli. So a small group of neurons can become activated, strongly activated by a very specific stimulus. And further, like the excitation of a circuit, excitation as being the final output of a circuit is not always like a good thing, so to speak. Sometimes the critical part is actually inhibition in a circuit to really like clinch things. So I want you to think about those three things, and you'll see how they come into play later on. So they start off by characterizing the behavior. In the mice, they show that the soon-to-be rescuers are not just displaying simple curiosity, because we know that mice are curious little creatures. So they put them in an open field with an anesthetized mouse placed in the center, and the bystanders will spend more of their time in the middle of the open field, inspecting their anesthetized buddy. And this isn't the case if what's in the center is a pretend mouse, a toy, or if it's a cadaver, like Vincent was mentioning earlier.
Oh, sorry, Vivian. I thought maybe I should reiterate like the players in the experiment.
Yeah, sure.
So these experimenters, I don't know, like Vivian said earlier, I don't know what got into them, but they decided to anesthetize a mouse. So you can give it an injection or give it some gas anesthetics, and the mouse would be out cold. It would still be breathing, it would still be mouse body temperature, but it's just asleep, not really moving. And then you put this mouse inside an empty cage called open field, so an empty box. And then you put a cage mate, so a friend, or it can be a stranger mouse. Apparently, it doesn't matter if it's a male mouse. Matters a little bit if it's female, but we're not going to get into that. But you could put his friend inside this open field, and this friend would run around, and it would go towards the anesthetized mouse. And the question that the experimenter is interested in is the interaction between the anesthetized mouse and the friend who's running around, and they call the friend the bystander mouse. And the bystander is going to engage in some very strange behavior, as Vivian's going to tell us. But yeah, so there are two players in this scenario.
Right, and as you could probably piece together from the title, the bystander's behavior, this tongue dragging, leads to a quicker emergence from anesthesia. So the bystander will do a couple of different things with respect to the anesthetized mouse. Like I mentioned at the beginning, sniffing, grooming, eye licking, and tongue dragging. And they'll spend most of their time sniffing and tongue dragging.
You'll get sued
if you
in
do
the US.
that.
If someone passed out and you lick their eyeballs.
Yeah. But, it's weird. It's around is like, eye licking is its own thing. I think there needs to be an offshoot paper about that, because it was kind of the odd one out for a lot of the things that they looked at. But in addition to just, if anybody's like watched videos of like behavioral assays with mice, they might be present in some of the papers that we've done in the past, then you can maybe appreciate that like with the lighting and the position of the camera, sometimes it's hard to know exactly what the mouse is doing. But, I don't know if they did this. I didn't look in the methods to see if they did this by hand, but I'm guessing.
They have video. So this paper is
Right. But did they do it did they like have, I don't know if they had somebody
like hand
scoring it?
I'm going to guess they did hand score it, because otherwise it would be very difficult to find like tongue dragging.
Right.
But,
well, which is why they do EMG, the electromyogram.
I guess I'll quickly highlight that this paper is open access if anyone wants to check it out. And they also have videos. If you go to the show notes, I'm guessing there'll be a link, and you can look at the video of these mouse behavior we're talking about. But yeah, sorry.
Yeah, no worries. No worries. But I mean, that's exactly that. I think people should look at these videos and then kind of ask themselves, could I reliably say what that mouse is doing with its face? And so they put these electrodes on the jaw muscle, and they can show that, like, even if you're not looking at the mouse, if you're just looking at the traces of muscle that are representative of muscle activity, you can differentiate sniffing, grooming, and the tongue dragging behavior. The final one being the one that shows the most activation of the jaw muscle.
And the tongue dragging, I'll quickly describe what goes on. It's actually quite weird if you watch the video. So you have your anesthetized mouse kind of like just slumped on the floor. And then its mouse friend would come along, start sniffing, and eventually it'll basically look like it's giving mouth to mouth. It looks like it's giving mouth to the mouse. But then you see it's actually obviously not blowing into the mouth and not doing CPR. And it's actually picking up the tongue of the mouse that the other mouse that's asleep, and then kind of like pulling the tongue a little bit. It doesn't look like vicious. It's not like,
just
not
tugging.
like a dog. It's just tucking it. It's not like shaking its head and dragging it around. It's just pulling
like
it
a dog.
Yeah, exactly. Like it's not doing anything that looks like it's actually trying to hurt the other mouse. It's like kind of pulling it out a little bit. But the researchers in the paper, they mentioned no one has ever described this before. So they propose it's a newly discovered behavior.
But I bet you it's one of those things that like people have seen. There should be a journal or a website that's like weird stuff that you saw. And you're like, what is this? We see those things all the time in cell culture. You're like, what is that? Like when you do this or that or the other thing. But it's never the question you're trying to answer. It's just like a descriptive thing that you kind of keep in your mind. But yeah, if I saw this behavior, and I don't do behavior with mice, but if I've seen it before, then it's like, oh yeah, it's probably happening consistently. Every time we take mice out to, for example, infect them with a virus or something like that, then we have an opportunity to see it happening. So Tim mentioned before that there is a little bit of a sex difference. So they found that the male mouse doesn't care if the bystander does not care if it's a male, whether the conspecific is familiar or a stranger. But the females the males will still engage in this tongue dragging behavior, but the females are more reticent to perform it on mice that are strangers. So they have a familiarity preference. So to kind of remove that variable, they do the rest of their experiments just in males. And so what's funny, actually, I think I'm like, somebody went to, BioRender, the website where you can create these beautiful figures and that like everybody's using these days. I swear that they like went to BioRender and they're like, can you make a figure of a mouse like doing body jiggles? Because if you look at the first, at one of the schematics, they show the tongue dragging is associated with this hind limb and tail and body movement trembling in the anesthetized mouse. Which although
I didn't see. I don't know whether they've verified. No. I don't know whether they've verified that by EMG of the asleep mouse. Because they say on the video it looks very obvious. They say on the video it looks obvious, but I looked at it and I was like, maybe. I don't know. Yeah.
So I didn't look either, but I don't believe that they did. It's not. If they did, it would be in the supplementary data.
In the schematic, you see the asleep mouse wiggling or like shaking as its tongue is being gently dragged.
See, I'm going to try this. Actually, no, our protocol doesn't allow me to pinch tongues, so I can't do that. But so they're thinking, they're like, what if this tongue dragging behavior is a way to try to rouse the anesthetized mouse? And they looked at how quickly mice that had their tongues pulled on, how quickly they returned from anesthesia. And it was actually faster than in a condition where the anesthetized mouse did not have a bystander present. And so a lot of the things that they're going to end up doing later on, it's difficult, or would be difficult to do them when you have two mice. In one situation, they need, or in one set of experiments, when they're mapping the circuit that's happening inside the bystander, they need the bystander to be there with the anesthetized mouse doing its thing. But when they're mapping out the circuit that's happening in the anesthetized mouse, it's easier to do some of their manipulations if they're able to kind of activate whatever is being activated by the tongue dragging without the bystander present. So they do, they pinch the tongue with some forceps, and they also do the same thing with the hind paw and the skin on the back. And they find that it's specifically the pinching of the tongue that brings the animals back more quickly. It's not like the presence of the bystander in some other way. It is really just having the tongue pinched and kind of tugged.
Which is just insane if you think about it.
Yeah, and it was also funny is that like in babies, I'm seeing a lot of babies, a lot of one particular baby these days, and babies when they suck, it's very soothing and they actually like fall asleep. So I was like, that's interesting. I wonder how that. Yeah, it made me think of that. I was like, well, when my baby is using their tongue, he passes out.
It's just got to pinch it instead of sucking.
Yeah, I guess.
Different modalities.
And in this paper, they didn't talk at all about like whether like nociception was involved or not. They don't go into that. So the ability to sense or bring pain stimuli from the periphery into the central nervous system. So they've characterized the behavior. We believe it's there. The bystanders are moved to tongue drag, and the tongue dragging gets the anesthetized mouse moving again. So they want to look at the circuits that are responsible for this, like I mentioned, in both the bystander and in the anesthetized mouse. So they're going to start with the anesthetized mouse. And here's where we get to the first of the 3 points or concepts that I want to convey. Like I said before, I realized as I was reading this paper that I really take for granted all these very cool and I think neuroscience-specific tools, like repurposing viruses, adapting them, and leveraging them to hop from one neuron to another and carry genes that are going to make the cell that they're infecting light up with a fluorescent protein, or they can control the neuron's activity either using light, optogenetics, or chemogenetically, which is like the newer kid on the block, using something called DREADS, which we'll get to later. But I don't know, have either of you seen these kinds of tracing tools present in other parts of biology? Not that you spend a lot of time reading.
Maybe working on neuroscience, but like
I don't know. Maybe Vincent would see something.
Yeah, I've seen these used in various papers. Yeah, especially AAV is a common virus to use for this with Cree. That's going to cut at a lock sequence. I've seen that before. I don't remember exactly where, but yeah.
Okay. Yeah, I think there are different components that.
But I think kind of mapping physical circuitry really is a neuroscience specific thing because it's really the nervous system that connects kind of everywhere in the body. That
I just wanted to say that I'm very neurocentric. For me, it's like the nervous system is the center of my universe. So I don't always think about it from the outside. Okay, so. So the first step in tracing this arousal circuit is to find out where the tongue-dragging stimulus heads to next. So they use a retrograde monosynaptic dye. So the retrograde part corresponds to, as you might expect, the direction that the dye goes in the neurons that have projections between the sensory organ, the tongue, and whatever brain or nervous system region receives those sensory inputs. So if something is anterograde, it means that the signal is traveling like an action potential does away from the cell body and toward the axon terminals. So retrograde is going the opposite direction from the terminal to the cell body. And the monosynaptic part is the number of like hops that the dye can make, the virus could make. And I don't know the specifics of how this works, but this restriction of being able to only jump one synapse is important because axons go all over the place. Neurons sometimes have bifurcated axons. So this dye kind of allows the experimenters to focus solely on the next step after the tongue. So they inject the dye into the tongue, where it gets picked up by what we later learned is called a pseudo-unipolar neuron. So that means that the cell body resides in one place. In this case, it's in something called the trigeminal ganglion. And the axon extends in two directions. Both of them are heading away from the cell body. But one end harbors the dendrites, and the other end harbors the axon terminal. So this is different than kind of like what we tend to think of when we think of neurons, which is the cell body with dendrites around it like a tree, and then the axon extending like the trunk of the tree. So the neuron will pick up activity in one place and then send an action potential, shoots down the axon, passes the cell body, so going through the trigeminal ganglion, and then continues on to the axon terminal. So the synapse, Tim, or somebody wanting to say something?
Sorry, I'm just wondering, so just because I got slightly confused by this when I first read it, but it's actually, these are cells that have... So are they similar to cells that the sensory neurons that are in the dorsal root ganglion? Are they also pseudo-unipolar
you mean in terms
cells?
Yes, in terms of how they are organized?
Yeah, because they are, because they have, what I picture is like essentially very long dendrites that go all the way to the tongue. And these dendrites have like sense organs in them so that when the tongue is, being touched or pressured or maybe even being bitten, it would send signals down the dendrite to the cell body and also to the axons to the next stage.
Right. Yes, but then it becomes kind of a semantic question, like how far do you extend when does the dendrite become? When does the dendrite become an axon? Kind of.
But like for example, like does it have the dynein and kinesin like only going, because dendrites would have both kinin and dynesin going both direction, but like I think in axons it is like anterograde versus retrograde is like very specific. So I actually don't know any of those.
That has to do with, I think we're getting into the weeds here a little bit.
We're getting into the weeds here. Let's talk about it offline.
It has to do with the properties of the channels that are allowed, But yes, the point is,
but it's similar to a sense neuron, like a sensory neuron that's in the periphery that is sensing the outside world. Okay,
right. And that first synapse, though, if I'm understanding this correctly, is really it's not like the first cell in that synapse is a neuron, right? It's actually like one of the tissue of the tongue itself that has mechanoreceptors on it and releases calcium onto the dendrites, I believe.
Okay. Got it.
Just to make that point clear, because I think that helps you kind of organize this daisy chain of cells.
So that's a synapse that is inside the tongue.
I think that's how I understood it.
And then gets transported back. Okay,
right.
Got it.
You have like a mechanoreceptor that senses the pressure on the tongue tissue, and then usually those, and that's kind of the thing for a lot of our sensory systems is you have a specialized sensory cell that will then it just generates a chemical signal, usually calcium, that it releases onto the neuron, and we know that releases it onto the dendrites of the neuron, and we know that that the calcium then I think will, trigger the action potential. But I'm not an expert, so I don't know. Okay. So in doing this, like injecting of the dye that can then kind of that can then go. Maybe I got this wrong, but this is kind of how I was envisioning it about the directionality. But they injected it into the tongue, so maybe circle back to all of this in the show notes or something when I make sure that I'm getting this right. But they find that the neurons that are receiving the sensory input from the tongue are in the trigeminal ganglion, and that their axons are ending in the brainstem in a place called the mesencephalic trigeminal nucleus, so the MTN. And this was actually already known. That is just a confirmation that the MTN is the only place that receives incoming sensory information from the mouth. So they've done this, they've taken one approach to show that there's this connection, but then getting to the second overarching concept, you have to you have to come at your observations, come at your hypothesis from multiple approaches. Maybe you have 2 or 3 or 4 ways of getting at the same phenomenon. So they have another approach where they use a retrograde transsynaptic pseudorabies virus that drives the expression of GFP, green fluorescent protein, And they observed the same thing they did with the dye, that the GFP co-localizes with, they saw the same thing as the dye, kind of mapping that same path, and that the GFP co-localizes with markers of glutamatergic neurons. And so then, just to be extra sure, they use one of these clever configurations of these mapping tools. So in the tongue, they inject a modified virus that drives the expression of a Cre recombinase under a promoter for a gene called synapsin, which limits the Cre expression to neurons. And then in the MTN, they inject another modified virus that has something called a DO sequence, double floxed inverted open reading frame, DIO, which when it's cleaved by the Cre, flips itself and allows for the transcription of a neighboring sequence, which in this case is GFP. So if you put all that together,
clever.
I know, right? So cool.
That's
and it
cool.
gets better.
That's very cool.
It gets better. Oh, it's so amazing. So you have this tongue-specific projection neuron that fluoresces green in the MTN and co-localizes with markers of glutamatergic neurons, but not with they do some staining for other cell types, and there's no co-localization with other neuronal cell types. Then they take yet another approach, because now we've got, I think that's 3 so far that they've done. So their 4th approach is they inject a virus that has that DO sequence. So when it gets activated by Cre, it flips and allows, in this case, the expression of a red fluorophore called M-cherry. So they inject that virus into the MTN, the brainstem, of a mouse that expresses Cre under the vGlut2 promoter, so glutamatergic neurons only. And then they take 2 pieces of tissue. They take, the injections are happening in the brainstem. So then they take the trigeminal ganglion and they take the tongue and they clear these 2 pieces of tissue. And the clearing process, I believe, is like it passes the tissue through a series of detergents, and this renders the tissue nearly transparent, and then you can image it using something called a light sheet microscope. And that just has to do with the direction of the light. So in a light sheet microscope, if you have the sample traditional microscopy, you have the light coming, in this Z plane. And it leads to issues that we can talk about at some other time. But light sheet has the illumination coming from the side. So you can take hundreds of single focal plane images, so like super crystal clear resolution, that when you put them all together, create this high resolution 3D image of the brain or of the tissue. And so
it's like a CT scan, but with visible light instead of X-ray.
Absolutely.
Yeah.
Because everybody knows how CT scans work.
Yeah. Well, I'm doing the dictionary trick. But we also previously talked about this like clearing technique. And the best thing about it is the name. It's called, in this case, before it was called iDisco. And in this case, it's a fluorescent of that. And they call it FDisco.
FDisco.
Is great.
That's so sad. Okay, so what's so cool then is that they show that they've turned stuff red by injecting it into the MTN, these neurons, and that red stuff is now detectable in the trigeminal ganglion, which is where the cell bodies of these neurons are, and also the tongue, which are like the dendrites that are receiving the sensory input from the tongue. Whew. Okay, guys, that's it. See you next month. That was a lot we already talked about. Like I said, this paper caught me on a good day, but there's more. Don't worry. There's more. That was only figure 2, and there's 6 data figures and a boatload of supplementary figures. But for the sake of maintaining sanity and attention and just time, we're not going to go through all of that again in each instance that they use it, but because I think my point has already been made about how clever this circuit mapping, the usage of these tools is. But I'm going to bring up in a couple situations things that are really cool.
But just to make it absolutely clear for listeners who might not have not be very experienced with e-circuit mapping, the trick is basically just to inject a virus that would cause the entire neuron to make a fluorescent protein. So you normally would inject the virus in the cell body, but you can also make this virus deactivate this virus unless it sees Cree, which is kind of like scissors. And you can inject the Cree at the terminals so that you can map the entire process.
Right.
And then once the neuron makes this fluorescent protein, the fluorescent protein goes everywhere of the axon, and also the dendrites and the cell body. And that, by tracking where this fluorescent dye goes, you can map out like where the entire neuron is, which is what we did. Yeah.
And you have to use light sheet microscopy to do these kinds of things because, or if you're going to be, imaging an entire, some of these neurons are huge, right? Even within the brain. And so it just doesn't lend itself to lots of other types of microscopy. So we're really fortunate to live.
Unless you have an army of undergraduate.
Yes. Ones that you trust. So it's really fantastic. But so previous studies have shown that these MTN neurons, so they're projecting or receiving information from the periphery, from the tongue, but then they're also projecting, so sending their axons to a place called the locus coeruleus, which is a brainstem nucleus that regulates arousal by releasing something called noradrenaline or norepinephrine into various brain regions. So they wanted to look at whether the MTN neurons are activated and then are activating neurons in the locus coeruleus during the tongue dragging event. So they introduced the bystander and the anesthetized mouse, and they let the tongue dragging commence, and then they collect the brain of the anesthetized mouse, and they look at the expression of something called CFOS, which is a gene that gets turned on very rapidly after a neuron is activated. And sure enough, they find that in the MTN, they're seeing, or in the locus coeruleus, they're seeing CFOS lighting up in the anesthetized mouse that were paired with the bystander, but that's not the case when the bystander is not present. So tongue dragging equals CFOS or neuronal activation in the locus coeruleus. But again, that's not enough. They actually turn to measuring locus coeruleus activity in real time in vivo with multi-electrode recordings after pinching the tongue and then pinching the hind paw and the skin on the back. And they find that all three interventions stimulate the locus coeruleus, but that the tongue pinch is way better at increasing the activity of that area of the brain than the other two pinch types.
And just a quick aside, the locus coeruleus is a classic kind of arousal system. Like
I wondered if I'd said that.
Yeah, so like people have done quite a lot of studies, like when you go from, but I'm not an expert, so I should have looked it up, but I can't. But if memory serves me right, as you transition from either REM sleep to non-REM sleep, or maybe when you start waking up, the locus coeruleus norepinephrine system start activating to allow you, like it's correlated with you waking up. So it's well known to be very important in kind of sleep-wake transition. And they also cited some papers where previously people found as you wake up from anesthesia, the locus coeruleus system is also, and the norepinephrine system is also engaged and important. So that's the reason why they are interested in the locus coeruleus.
Critical to mention that. And as I was talking, I was like, did I say that? I'm not sure. So thank you so much for making sure that was raised. Otherwise, it would make no sense, right? So I said that I'm not going to go into extreme detail on the next few things. But again, they're very clever and cool. But I'm going to say them slowly, and then people can, like, if they choose to think about it more deeply, they can. So they inject into the tongue this virus that expresses it's flippo. I guess it's just a flippase, but it's a virus that drives the expression of this flippase only in neurons using this synapsin.
It's just flippase. It's just like Cree. So Cree is like molecular scissors that recognize a specific sequence to cut it so that you can have like conditional expression. Right. And flippo is just a different version of Cree. So it's different pair of scissors that recognize its own special sequence.
So you can use them together in the same mouse.
What is known as technically multiplexing.
Ah, okay.
Or intersectional expression. There are different ways of
intersectional expression.
Yeah.
Don't use that word. The NIH might find us and shut us down.
Yeah.
Okay, so in the tongue, they inject this retrograde synapsin flipovirus. And it's going to target the second of two viral constructs that I'm going to mention that are injected into the brainstem, into the MTN. So into the MTN, they have a mixture of this DO-M cherry virus and an FDO channel rhodopsin GFP virus. And so only that second viral construct is going to be targeted by the synapsin-driven flipase. And then this is happening in a dopamine beta hydroxylase Cree mouse. So that Cree is going to work only on the DOM cherry. So, and the dopamine hydroxylase drives Cree expression only in locus coeruleus neurons. So then you've got green MTN neurons and red locus coeruleus neurons, and you can activate the green neurons, MTN neurons, through optogenetics. So they detect, when they do this, they detect excitatory postsynaptic currents in the locus coeruleus, the red cells. And this is done ex vivo, by the way, so they took a slice of brain and they put it in a dish and they're measuring things. And so those excitatory postsynaptic currents can be blocked if you add a glutamate receptor antagonist. So again, being like these are glutamatergic projection neurons from the MTN into the locus coeruleus. So again, we've got to do a double, triple, quadruple tap to make sure But this is a
kind of neuroscience technique that is increasingly available to us, and it really helps us map the circuitry.
It does.
Making only the locus coeruleus norepinephrine neurons glow red so you can find them under a microscope and only record from them. Be absolutely sure you're not recording some other cells. And then only making neurons in the MTN, but only the subset that projects to the tongue. Only those that express channel rhodopsin. So when you shine a blue light, they can release glutamate. So that you can record from your locus coeruleus ret neurons to detect a current. This kind of technique is becoming more bread and butter. And it really
I just
helps
lust after these things, these viruses.
Yeah.
I'm like, these constructs. But I want to take a minute to say that troubleshooting this stuff, doing all the work ahead of these experiments to make sure that everything is working the way you expect it to, is so important. And I really want to emphasize to anyone who's listening that you really, if you're doing science, please take the time and the mice and the resources to make sure that you are actually doing what you think you're doing. Things are working appropriately. And look for evidence of that in papers that you read.
I can attest to that because our lab I've been trying to pilot retrograde AAV retrograde Cree, like this paper has been doing. And when we injected it into the striatum, it just killed all the cells. So you got to really pilot, and it turns out the dilution is very important. So there's a lot of troubleshooting in the background, like everything in science. You got to fine tune your parameters.
Yeah. Okay, so here we've got this beating a dead horse thing that I said before. So we really want to make sure that this MTN locus coeruleus functional connection in the context of tongue dragging is a thing. So here they're going to use chemogenetics as opposed to optogenetics to test that MTN neuron activation leads to locus coeruleus neuron activation. So in the tongue goes that retrograde synapsin Cree virus. In the MTN, we have a Cree-dependent DO HM4DI M-cherry. So that HM4DI, which I might just say 4 or DI, maybe I'll just say DI. I don't know how people talk about this, but
I think people just call it inhibitory
dread. Okay.
But then we have to explain what dread is.
Dread means designer receptor exclusively activated by designer drugs. And so this inhibitory dread inhibits neuronal activation in the presence of a drug called CNO, clozapine N oxide. So they say in here, we confirmed that this system works ex vivo, and then we tested it in vivo because they're going to use it multiple times. So with this inhibition of the MTN neurons, they find that the CNO treatment, so it causes this inhibition. It blocks that speedy return of the mice from anesthesia, which, by the way, I don't think we mentioned it before, but they're measuring the return of writing reflex. So I think, Tim said like the mouse is all like slumped over when it's anesthetized, and then it starts to like try to come back, and it goes from its side or its back onto its feet. So that's the writing reflex.
It's weird because a lot of humans I know, including myself, do not have a writing reflex.
Why, you just open your eyes and like stare at the ceiling or something?
Just leave me lying on my back, please.
Yeah, with all of my squishy bits,
I don't know whether
like
it's
exposed
a true reflex
to predators. I don't know, but it is in. I feel like a lot of dogs, cats,
animals do have writing reflex. Yeah,
yeah,
guess so.
I mean, we're animals too, but So what they do is they do their tongue pinching. So like the bystander mouse is not here, right? So they're doing the tongue pinching. And if you inhibit the MTN activation, that like speedy writing reflex, return to the writing reflex doesn't happen. And then conversely, if you use an activating dread. Which I think they didn't say it, but I think it was actually did they just use the activation and not, or like was it in the absence of the tongue pinch?
I think so.
I think so, right? Because
otherwise, I remember correctly,
yeah,
they just injected it, and I can't remember if they paired it with. So they gave the activating dread. And then they shoved a bystander mouse in. So the bystander would still go and try to tongue drag a little bit, but each tongue drag has an exaggerating effect. So the sleeping mouse recovers faster, something like that.
Yeah. Okay.
So the inhibitory dread kind of, in my mind, it works almost like a local anesthetics. Like if you can't feel your tongue, then your helper mice is not going to be able to pinch you out of your slumber. Right. That's what I'm thinking. Yeah.
And so here they are actually, as opposed to you, Tim, who you're trying not to kill your cells, they actually do try to kill their cells. They specifically ablate glutamatergic neurons in the MTN by infusing that area of the brain with this retrograde synapsin flipovirus in the tongue, and then a flipodependent genetically engineered caspase-containing virus. So even when the bystander performs the tongue dragging, the anesthetized animals have a latency to return, like to awaken from their slumber that is comparable to that of mice that didn't have the constructs.
And the caspase acts as a way. So this is a new experiment that Vivian just described. So Vivian mentioned they like to triangulate their experiments. So they try to test it in different angles. So this is a new experiment where they killed the cells using caspase, which triggers apoptosis. So like programmed cell death. So they showed that once you kill the MTN, then the tongue dragging is less effective. Yeah. So once again, implicating the MTN circuitry.
Yeah. Very cool.
Yeah.
All right, so I think we've pretty much figured out what's happening in the anesthetized mouse. So now let's move to the bystander. So figuring out where to start mapping this circuit is a little bit trickier because we don't actually have any indication of where it's starting.
Actually, yes. I wonder, so what happened to the anesthetized mouse they proposed is that the tongue is being pinched either by the experimenter using forceps or by a bystander-friendly mouse, the tongue signal goes to the MTN, and then the MTN talks to the locus coeruleus, the LC, and the locus coeruleus triggers, releases norepinephrine, and then that wakes the sleeping mouse up. But they never, we're doing nitpicking kind of online podcast reviewing here. I think the thing that they didn't do is actually block the locus coeruleus. So they never stop the locus coeruleus from firing to show
Here,
that it is.
I'm biting my tongue and seeing if anything happens. I don't think anything happens.
But Vincent, you have to put yourself under anesthesia first.
I'm not asleep, but I was wondering if something would happen that.
But maybe it's one of those things that if you do it to yourself, it doesn't work.
Yeah, I was just going to say you can't tickle yourself.
And it has to be someone else doing it to you.
No doubt.
If the sensory receptors are there, it should work, right?
No, but if you have like with the tickling thing, if you have some like representation in your mind of what it's going to feel like if you do it, because like it doesn't work out.
You might actually inhibit the sensation.
Yeah, exactly.
Anyway, we're highly speculating here.
Yes. Returning to less speculative topics. Like I said, it's a little bit trickier to determine where to start when we're mapping out what's happening in the bystanders, because like with the arousal circuit, we know that it starts with the tongue. So we're obviously going to start there. But so here they do kind of a screening, again, using CFOS. So they put the bystander in there with the anesthetized mouse. The bystander engages in the tongue dragging activity. And I think there's a couple of little like nitpicky things that I could talk about here, but I'm not going to. About the timing and the other things that the mouse did prior to tongue dragging. But they put the bystander in, the bystander does the tongue dragging, and then they collect the brain and they look for CFOS. And I think they were tipped off a little bit by previous literature to look at something called the paraventricular nucleus of the thalamus and the shell of the nucleus accumbens, because those two brain regions have been implicated in motivated and consoling behavior. So I'm guessing that's why they didn't go through like the whole brain, but rather decided to look at those brain regions because of the previously published rationale for their potential involvement. And so, they found that in the PVT and the nucleus accumbens, there were high levels of CFOS. In the PVT, the CFOS was showing up in like almost or more than 95% of the glutamatergic neurons there. And it was also showing up in almost 90% of the GABAergic neurons in the nucleus accumbens. So we've got these glutamatergic neurons in the PVT getting activated, and then these GABAergic interneurons getting activated in the nucleus accumbens. So those places are probably involved in the circuit. So, and
just to reiterate, this is in the mouse that is in the friend mouse that is trying to rescue its sleeping friend.
It's the bystander, the Good Samaritan.
Bystander mouse,
So they implement the same approach, many of the same approaches that they used for the previous circuit. They map they do retrograde tracing of projections that are coming from the PVT into the nucleus accumbens, and they find that the nucleus accumbens neurons, which they've labeled with M-cherry, have terminals in other regions, which we're not going to really go into, but those other regions are the anterior hypothalamic nucleus, one of them, which has been associated with biting attacks behavior. So I thought that was interesting. So now they're gonna look at activity in the brain in these brain regions, but they're gonna take a slightly different approach than what they did previously, because in the first circuit, the animal's like knocked out, or they're gonna be taking it's not doing anything. It's just stuff is happening to it, and so they can also look at these things outside of the living animal. They can look at it ex vivo in a brain slice in a dish. But here, they need the animal to be behaving and acting for them to measure things. So they're gonna measure neuronal activity using these genetically encoded calcium sensors, because there's calcium activity, kind of calcium activity, neuronal activity. That was really hand wavy, but that's what you got to know.
Basically, when a neuron fires, calcium goes in enormously. And so if you have a dye that is sensitive to calcium, which people have designed, then you can look at when this dye glows stronger as a proxy measure of the neuron firing.
So using this, they show that there are these massive increases in calcium activity in the PVT when the bystander is performing sniffing, grooming, and tongue dragging, but not eye licking. See my previous comment about needing a paper on eye licking. So then, but what happens if you prevent the animals from performing the tongue dragging, or what if you drive the activation of that circuit, or like essentially we're saying suppressing or promoting, but you can't like totally stop something from happening or totally make it happen. So they're going to use their inhibitory and activating dreads injected in the PVT of an animal that has Cre driven by the CAM kinase 2 promoter. So that's an enzyme that's in glutamatergic neurons. And then they're going to infuse the CNO, the drug that binds to those dreads, to either inhibit or activate this PVT nucleus accumbens circuit, respectively. And seamlessly, they find that these manipulations, so the inhibition of the PVT or the activation of the PVT, they reduce and increase the tongue biting or tongue dragging behavior in the bystanders. So we're getting close to the end here. Just an interesting little twist. They use these genetically encoded calcium sensors in the nucleus accumbens. Using them, they show that when the bystanders are engaging in the other behaviors, there was a lot of calcium signaling happening in the nucleus accumbens. So like excitation of neurons there. But when the animals performed the tongue dragging, there was actually a decrease in calcium activity. So some inhibitory neurons are like between the PVT and, we'll get to it later, but these dopaminergic medium spiny neurons, which I'll get to at the end. It's
a bit tricky.
Yeah.
Sorry, I don't know how much you want to go into it, but the idea, I guess we're diving in. The idea is that the PVT, which is a nucleus in the thalamus, is releasing glutamate in the nucleus accumbens, part of the ventral striatum. And glutamate usually excites cells. So it makes sense that when the PVT is firing during sniffing and grooming, you see the nucleus accumbens also firing kind of together because it's activating the cells. The one thing that doesn't make any sense that the researchers are going to have to explain soon, that we're going to get into, is why does PVT, during tongue dragging, the PVT is actually firing. So they found that in a previous experiment. PVT fires, but the nucleus accumbens, even though it's receiving glutamate, is actually getting inhibited. So that's one crucial thing that we're going to have to find out.
Yes.
And Vivian's going to tell us how.
I am. So they know already in this, like in a PVT striatal circuit, that the PVT activation recruits these parvalbumin expressing interneurons. So inhibitory interneurons. I mean, I think aren't all interneurons inhibitory, right?
They can be in the striatum. They can be cholinergic. So they're actually releasing acetylcholine.
Yeah.
Yeah. Okay. So they're also interneurons are weird.
Okay, great. I just wanted to know that I wasn't
inhibitory.
Yeah. Okay. So it is worth saying inhibitory interneurons, just to be precise. So
yeah.
So knowing this, they posited that maybe it's parvalbumin expressing interneurons in the nucleus accumbens are the targets of the PVT glutamatergic inputs. So they image calcium activity at the this is crazy, single cell level. I'm not going to go into how they do this, but check it out. It's mind blowing. Single cell level calcium imaging in the neurons in the nucleus accumbens that are receiving inputs from the PVT. And they find that for all of the behaviors, so sniffing, grooming, eye licking, and tongue dragging, the nucleus accumbens neurons kind of fall into three groups, either increased calcium activity, decreased, or non-responsive. And here's what they say. Although fewer nucleus accumbens neurons were activated during tongue dragging behavior, the calcium activity in these neurons was higher than during the other behaviors in bystander mice, implying that a small proportion of PVT innervated nucleus accumbens neurons may specifically respond to rescue-like behavior. And this was my third and final point. Bigger is not always better. More is not, or more is sometimes less. And excitation as a final output is not always necessary for recruiting a circuit or activating or using it in such a way to achieve a certain behavioral outcome. So I think it's important to not just that more activation is better or more neurons is better. Like we got to root for the underdogs too. Sometimes.
Yeah, I think the theory here is still quite speculative from the researchers' point of view. So they're saying what happens is during tongue dragging, that when the bystander mouse is performing the tongue dragging on its sleeping friend, the PVT neurons is preferentially activating these inhibitory interneurons in the nucleus accumbens. And these inhibitory neurons' job, they are interneurons because they basically only talk to cells within the same region. And because they are inhibitory, they inhibit other cells. Mm-hmm. So basically what happens is when the mouse is engaging in tongue dragging, a very, very small amount of cells, these inhibitory neurons light up. And that's 12%. What you're seeing using calcium imaging. And 12% of them light up. And they are suppressing everyone else. And that's why you see a dip in the overall activity. And what's interesting, they also show that these subset of neurons, when they do light up, they actually have very big peaks. So the amplitude of the calcium signal is higher, but they don't light up very often, and only very few of them light up. So that's the hypothesis that they propose to us, that these parvalbumin neurons, parvalbumin positive, so it's just a cell marker. These inhibitory interneurons very important for the tongue dragging, which they're gonna, if you have time, they're gonna try to prove to us in the next experiment.
Yeah. So we're so close to the end here. So so they make the distinction that it's parvalbumin expressing interneurons, not somatostatin expressing interneurons. That was meant for a different population of neuroscientists than me. So have fun.
Yeah, that's definitely in the weeds.
Yeah.
Splitting hairs.
So here we're down to the last two things, I think, or last 2 or 3 things. But so another approach that they used was ex vivo, so brain slice in dish. And it involved making the glutamatergic PVT neurons red and sensitive to blue light. So that's using a virus that drives channel rhodopsin and mCherry under the CAM kinase 2 promoter. And then the parvalbumin positive nucleus accumbens interneurons, green, so that's DO-EGFP in parvalbumin-Cree mice. And when they record from the green cells in the nucleus accumbens, following channel rhodopsin-mediated activation of the PVT neurons, they can reliably detect these excitatory postsynaptic currents. And then if you can block that by blocking glutamate pharmacologically, the glutamate receptors. And so then to test that these parvalbumin interneurons, that their activation and then some, like Tim was saying, inhibition of downstream cells is necessary for the tongue dragging behavior. They use dreads to either inhibit or activate those interneurons. And when they reduce the interneurons' activity, the tongue dragging behavior took longer to manifest, as did the duration and frequency of sniffing. And then conversely, when they activate those interneurons, the tongue dragging behavior happens more quickly. And it also increases the duration and frequency of sniffing. So I'm thinking that sniffing is probably like a gateway to, it's a necessary component probably of activating the tongue dragging behavior, but it can exist on its own without developing into tongue dragging. And so then using all of those techniques that we've already talked about, and like the back of your hand at this point, they determined that the downstream target of these interneurons in the nucleus accumbens are dopamine receptor 1 expressing medium spiny neurons. Big reveal. And it wasn't the dopamine, the D2 MSNs of the nucleus accumbens. So, boom, we're done. I think everybody needs to go rest their brain now because that was quite a trip.
I have a question. So in the bystander mouse, does that mouse have to see the other one that's anesthetized, or does it sense it? by smell?
Interesting.
They didn't do any experiments.
They have to do it in the dark, but
No, they can see anyway, can't they?
That if they do a complete darkness, then surely they can't see.
I don't know.
Do we need to have blind mice?
3 blind mice.
I mean, this PVT that gets activated, is that dependent on vision or?
I mean,
no,
the sniffing, the fact that the sniffing preceded or like changed with in that last set of experiments, changed with the latency of the tongue dragging, suggests to me that they probably need the sniffing somehow. Whether it's really necessary to trigger it, I don't know. But I think there are drugs that you can stick up into the nose that causes the epithelium to like slough off, so you can create this kind of transient loss of smell. So there's potentially a version of these experiments where you administer that first to the bystanders and then see what happens. I like that. That's a good idea. We should tell them to do that.
Yeah, it's possible that you both either vision or smell can do it.
Mm-hmm.
Yeah.
But the smell one's a lot easier, unless you make little masks.
Yeah, I was thinking of a mask but they'll take it off. They won't like it.
No, just turn the light off. It's okay. I mean, a lot of behavior experiment is done in the dark, just because the mice are less scared when they're in the dark than when they're in the light.
They are nocturnal, right?
Yeah, they are nocturnal. Yes. But I think it's easy for me to rule out vision, because I was like, mice are supposed to have very poor eyesight and they do a lot of things by sniffing. But a long time ago, when we talked to Iona Kasha and Rob Fromke, we talked about how a babysitting mouse can observe a mummy mouse behind like a glass window to learn how to babysit. So they can definitely learn by observation as well. So
it's just this idea that the mouse is looking, he or she sees the knocked out mouse. They put 2 and 2 together. It's just.
But I think if it's quite atypical for a mouse to find a sleeping mouse that wouldn't wake up when it tries to like lick its eye or bite its tongue. And I think it's quite easy to find its mouth, because you can tell because the mouse is breathing, perhaps.
So
then on the other end,
they do a little discussion. They do a little discussion of whether this happens in the wild, because in the wild, of course, there's no anesthesia, but mice do sleep. So are we saying that mice wake each other up all the time?
Well, I think right? I don't know. So getting back to that NPR story, I heard, I think potentially in a previous publication or a different publication or something like that, the experimenters found that this tongue dragging behavior could actually dislodge things from inside the sleeping mouse's throat. So I'm like, is this like the mouse version of, you're at a restaurant and the guy's choking on a piece of steak? I don't know.
That's interesting.
Heimlich maneuver.
Right.
Yeah.
Very cool. Anyway, that was brilliant, Vivian. I loved it. It was a great paper. Very interesting.
Yeah. Very
amazing
interesting.
Technology. Thank you. Thank you so much.
And a lot of work for the experimenter. They have to figure out the neurocircuitry twice, one in the sleeping mouse and one in the bystander mouse.
Exactly.
It's so much work.
Exactly.
Yeah, it's a great paper. I really enjoyed it.
That is TWiN 58. Show notes, microbe.tv/TWiN. If you have any questions or comments, TWiN@microbe.tv. And we'd love your support to do these programs, microbe.tv/contribute. Vivian Morrison's at Tulane University. Thank you, Vivian.
Thank you.
Tim Chung is at New York University. Thanks, Tim.
Thanks, Vincent. Thanks, Jen. It was a very fun paper to read.
Does anybody want to go eat some Mike and Ike's or some red and green M&Ms? Because there's so much red and green in this paper that we have to go eat some Christmas colored M&Ms.
I didn't get that, but I was wondering when will we have actual human surgeons start putting pinching a anesthetized patient's tongue into the protocol to wake them up. Maybe soon.
Very interesting.
Maybe.
I'm Vincent Racaniello. You can find me at microbe.tv. You've been listening to This Week in neuroscience. Thanks for joining us. We'll be back next month.